From the American Society for Microbiology, this is TWIV, This Week in Virology, episode 444, recorded on June 3rd, 2017. Hey everybody, I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Today we're coming to you from New Orleans, Louisiana. We're at ASM Microbe 2017. And we're in a new venue this year. We're in the exhibit floor. This is a huge warehouse-like space where all the exhibitors have booths. There are lots of people wandering around. So you're going to hear a higher noise level than we've had in previous TWIVs, but it's the way it is here. We have lots of people milling around. And joining me today, all the way to my left, Rich Condit.
Howdy there, Vincent. Here we are on the road again. This is good.
On the road, that's right. We have to get a t-shirt with all our road tours on the back of it, right? What's the weather today here in New Orleans?
I just looked it up. You know, we're inside here. The weather is inside. No, it's 79 degrees outside and partly cloudy here in New Orleans. That's 26 Celsius. Good.
No, it's not 46 Celsius. No
26.
way. 26.
26.
Yes. 46 would be
Light.
we'd be very hot. All right, we have a very special guest today. She is a molecular biologist, a virologist. She's from the National Aeronautics and Space Administration. She's astronaut Kate Rubins. Welcome to TWIV.
Thank you. It's great to be on the podcast today.
And I heard you've heard of TWIV before, right?
Yeah, absolutely. I try to stay in touch with a lot of different, kind of current happenings in molecular biology and virology,
right.
And keep up to date as much as I can in the NASA world with what's going on in science.
So I have to ask you, have you ever listened to a podcast in space?
I actually have listened to podcasts in space. We can do, when we're working out, when we're running on the treadmill, we can listen to some music, we can listen to podcasts.
All right, good to hear it. Now, I love your handle on Twitter, Astro Kate, and it's got a 7 after it.
Yes.
Is that because of the 7 original astronauts?
It does, and also the other, Astro Kate was taken when I tried to get my Twitter handle.
Okay, I just love it, Astro Kate. Have you ever been to ASM Microbe, by the way?
Yes, I had a long time ago when I was actively working in virology.
Cool, yes, go ahead.
What's the weather like on the space station?
Space station weather is always about 72 degrees, depending on the commander's preference, low humidity, and very little variation.
But you have day and night, or do you?
Well, we actually go through 16 different day-night cycles. So we have sunrises and sunsets about every 90 minutes outside. Wow. So we actually put the astronauts on an artificial time scale just so that we're not disrupting circadian rhythms, and they pick GMT, so we're actually European time, which is a nice even split for the US and the Russian control centers.
72 degrees, just like San Diego all the time, right?
And it never rains up there.
It never rains. I want to tell everyone in the audience, if you have questions, there's a microphone in the back. Also, if you're listening online at Facebook, we're streaming this. You go to facebook.com/ASMFan, and you can type your questions in, and we'll try and answer them here. Do you use, by the way, here on TWIV, we have always an argument between Celsius and Fahrenheit. Do you use Celsius or Fahrenheit in space?
So we actually use Fahrenheit if we're talking to the US flight controllers, and Celsius for everybody else.
Figures, right? Good figures. All right, let's start with a little bit of your history and learn where you were brought up and educated. Where are you from originally?
So I'm from California, and I went to undergrad at UC San Diego, so I can verify the weather is fantastic down there. I did my grad school at Stanford in the labs of Pat Brown and David Relman, and we worked on gene expression in animal models of smallpox and also Ebola and other filoviruses. And then after grad school, took a position at the Whitehead as a Whitehead Fellow and started my own research lab. That was chugging along merrily and going very well. And I saw astronaut applications one day and decided to throw my name in. And didn't really think anything too serious would come out of it, but I started getting some phone calls from Houston a few months later and ended up being selected in the 2009 astronaut class.
How long did the selection process take?
It's about a year, so we're actually just wrapping up our current NASA selection process, and next week on June 7th, we're going to announce our new astronaut candidates.
So do you do this every year, every few years?
It's about every 4 years, every 5 years on average.
And how big is the core at the moment?
So right now we've got 44 active astronauts, and we're hiring a small class coming in.
So I was thinking about this, now that you're in the Corps, are you involved in the selection process?
We are involved in the selection process. Everybody's kind of involved in the selection process to some extent. I wasn't on the selection board this year because I was in space when they started.
I was wondering what you look for. And do you have any idea why you got picked?
Yeah. So that's actually, I get that question a lot. And I think they're looking for people that are coming in from a variety of different tracks, and certainly the scientist track is one of them. It's not just, biology is actually a little bit rare. We're looking for physicists, engineers, and then there's also kind of the fighter pilot track, the military track. So it's a mixed civilian and military. NASA's a civilian organization, but we take folks from civilian and military tracks. And we're looking for folks that have, really strong demonstrated research records, but also people who have done things outside of just their research career, things where they would be in a remote, isolated, confined environment. So folks that have done field work are often very appealing to us.
And you had done some of that, right?
Yeah, so as part of my lab, we had a collaboration with the Army in the Democratic Republic of Congo. And so we went there off and on to look at monkeypox as it was occurring, and it's one of the very few places in the world that it naturally occurs.
In fact, you know Rich Condit before, didn't you?
Yep.
Well, we both worked on pox viruses.
Right.
Matter of fact, more specifically, pox virus transcription.
Right. Yeah, he tries to talk about that on TWIV. We don't always let him. So in college, you were a science major already, right?
Yes.
So, and then you got a PhD in science, did a postdoc. Where did the interest in science come from?
So when I was a kid, I wanted to be an astronaut, a biologist, and a geologist. Not in order, but all three simultaneously. And was pretty sure one could have multiple careers like that. Which was why geology training was such a kick. They actually brought some of the Apollo astronauts in to train us in geology, so that was great. And it's actually fun to see how much microbial work is now interacting with geology. But I thought, I'll do all three of these careers, and dropped the astronaut thing when it didn't seem like an actual career that real people did for a living around high school.
Yeah, that usually happens, right? A lot of kids want to be astronauts, and then reality sinks in.
Yeah.
Right? But now, what is a Whitehead Fellow? Tell us that kind of a position. It's kind of special, right?
It is. So there's a few places that do this. Essentially take people right after their graduate degree. You skip a postdoc and you start a small lab. So you get some research funding, and then you have principal investigator status that allows you to apply for NIH funding with PI status. It's a really interesting program. It takes you at a very young age and puts you in the PI position to be responsible for a lab. But I had a very clear idea of what I wanted to do, and there wasn't too many other folks out there doing that, and I really wanted to get started with setting up the lab and continuing some of the grant funding we had at grad school. So it's a great experience, I think, if you really know what you want to do. But I think the value of a postdoc is also extremely important too. It gives gives folks a chance to learn a little bit in another lab.
We had a Whitehead Fellow on TWIV, Gabriel Victora. Do you know the name?
Yeah.
And he's now at the Rockefeller
Yeah.
as well. But you're in space, and I actually had wanted to talk about some of your virology work, and Rich said, no, we do that all the time on TWIV. We're going to talk about space.
Yeah, we don't get to talk to astronauts every day.
So
right?
we would like to talk a little bit about space science,
Okay.
right? Because you're known for doing experiments in space. And I wanted to know if, when you were applying to the astronaut program, did you have in your mind to bring to it an experimental bent?
Yeah, absolutely. We're very operational in the astronaut corps, and you have to be. You're at the cutting edge of essentially a huge amount of energy. You're on the explosive end of a rocket, or you're living in a metal hole that's just a couple centimeters away from absolute vacuum. And so we tend to be extremely operational and extremely procedural. But at the same time, you're in an area where you are absolutely at the edge of exploration. Just the fact that we are constantly in free fall is mind-boggling. And we've had limited opportunities to do this lab work in space but over the years, but I really wanted to bring a complete research perspective to this. What are things that we can find out about human physiology? What are things about cellular or molecular biology that might be totally changed by being either in the radiation or the microgravity environment of space?
And was this something you pitched to them during the application process?
No. In the application process, you just need to be a trainable, trainable astronaut.
So how long did the training take?
So the initial training is 2 years.
Give me a. You applied when?
So I applied in 2008. It's about a year-long selection process.
Okay, and you were accepted in 2009?
Accepted in 2009, yeah. And then it's a 2-year training process. So there's a lot of things that you come in, skills you need to acquire. So you come in and you need to be something approaching fluent in Russian by the end of a couple years.
I was wondering about that.
So you start language training immediately. They take folks with no aviation background, put you in a T-38 supersonic fighter jet trainer. So I got the chance to actually go a couple times to the Navy to flight school to learn how to fly, initially in the back seat and then in the front seat of a T-6.
So you can fly that now?
So I can fly T-6.
Wow.
We fly T-38s at NASA. They're a little bit faster. It's excellent training for how to deal with an emergency situation. These are fighter jet trainers, so they I would say, they'll put you in a position where you're having to think and react much more quickly than you would deal with commercial aircraft. You also need to learn how to do things like spacewalking, which is not, this isn't a skill you pick up.
You do that in the pool?
You do that in the pool. So
you'd been in a spacesuit before.
Well, I'd been in a BSL-4 suit, so I came in with a little bit too much confidence there, thinking, yeah, this is no problem. I've been in a puffy white thing that's called a spacesuit. And the BSL-4 suit's about 30 pounds, maybe maximum. The spacesuit is 300 pounds. So
wow.
There's a bit more work. You're also, you're floating. You're in 6 degrees of motion, and you are crawling around on a mock-up of the space station. So there's a lot to learn there. We need to learn how to maneuver the robotic arm. So this is maneuvering a robotic arm. Also, again, that flying motion of all 6 axes and controlling the robotic arm. So it's a lot to learn. That takes about 2 years. And then once you get mission assigned, it's another 2 years for your specific space mission.
And what are you doing in the meantime?
So you're working in mission control. You're the capsule communicator. We've had that since the Mercury Gemini days, where there's always an astronaut or a capcom in mission control who's actually talking on the loops to our space vehicle. We're doing a lot of the training. We're participating in some of the development and design of the mission. So I did a lot of work trying to work on the rodent habitat that we actually developed for space station, to have an animal facility on board for some of these microgravity studies.
So there's a mouse facility on the space station?
There is a mouse facility on the space station,
I can't imagine what the daily cost is for that, because we pay about $5 per cage per day at Columbia. It's probably orders of magnitude.
It's expensive. The mice also get selected for launch as well. There's a selection process there.
So it seems to me that there must be some experiences in the actually going into space in the space station that they can't possibly simulate, okay? So what, were there surprises?
Yeah, absolutely. We try to train as much as we can on the ground, but we're constantly encumbered by the fact that we live in a 1G environment here. And so there's some things that you can do in the water that will help you a little bit, but for example, when you're out on a spacewalk, you're in this 300-pound suit, but at the same time, you have no control over your motion. So in the water, when we train in the pool, we always have water drag, which stabilizes you. On board in a spacewalk, you're completely unstable, yet you're hanging on to the outside of the space station in absolute vacuum going along at 17,000 miles an hour. So there's not a lot that we can simulate really on the planet that will train you for that. Part of it is just being in situations where unexpected things pop up and you have to deal with them, and then you have a confidence that you can deal with future unexpected situations.
So is part of the selection process that you need to be reasonably physically fit? Is that?
Yeah, you definitely either need to enjoy spending time in the gym or be able to get yourself into shape fairly quickly. There definitely is a physical component to the process. On board, we exercise about 2.5 hours a day just to maintain our muscle growth, and it actually is one of the best countermeasures against bone loss that we have, resistive exercise. So we need to be trained and in good enough shape to handle kind of a lot of the physical rigors of launch and entry are quite severe also.
So 2.5 hours of exercise a day, plenty of time to listen to podcasts.
Yes, absolutely.
So do you have any trouble falling asleep?
So some of the pressures of working up there, those can be difficult. This kind of being a very artificial environment, 16 sunrises and sunsets, and so you actually have to think pretty carefully about making sure that you're not going out and looking at the Earth right before you go to bed, right? This is the most amount of blue light that you could possibly imagine. It's this huge light source. And you have to, it's such a high-paced, high-tempo environment. You have to be a little bit careful that you get all the work done for the day, but you have got to sleep, because you have to be ready to be woken up by needing to fight a fire in space at 2 in the morning. And so you can't, you have to be able to push it, but you can't ever be doing 3 kind of all-nighters in the lab type thing, because you have to be ready for an emergency at any time.
So you can fall asleep without an issue then?
Yeah.
Close your eyes and that's it?
Well, kind of calm down from all the exciting things we've been doing.
I can't imagine. You strap yourself in and you're floating, right?
Yeah, so you actually we have a little sleeping bag in our crew quarters, and you can put yourself in the sleeping bag, and that's mostly just to keep you from floating away
around.
and bumping into a computer or a rack or something.
Or a mouse?
Or a mouse.
Can we move on to science?
Yeah.
So what was the first biological type experiment you did in space?
So getting up there, we actually had a SpaceX 9, a Dragon vehicle come, and that brought with it some live cardiomyocytes. These were IPS-induced cardiomyocytes that they'd actually plated out on the ground, launched.
So they're already on plastic, right?
They're in a very unique 6-well plate that then has a gas permeable membrane on top, so that the liquid doesn't float out, and a little lower lock port so that we could change the media. So my very first experiments were cell culture up there. I would have to say that was probably the most nervous I've been about a cell culture experiment, because you contaminate that one, you're not launching another one from the planet.
You can't go to the freezer, right?
Yeah, you can't just be like, well, that was a bad day, I'm going to go thaw a new cell line.
So what was the experiment there? Just see if you could do it?
Yeah, so we were looking to prove the feasibility of this long duration cell culture on orbit. We had a newly developed CO2 incubator up there, the technology for cell growth, but they were also looking at cardiomyocytes, gene expression, studying with video microscopy the actual beating to see if there was a change when you have these cells that are in free fall, if they're going to change their 3D structure, their communication with each other, and their gene expression.
So you were on the station for how long?
So I was there for 115 days.
Okay. You can do a lot of experiments.
4 months.
Yeah.
And were the cells there the whole time?
They were there for about 30 days. So they launched, we cultured them the whole time, and then took some, fixed them, put some in RNA later, and sent some back down on a SpaceX, which landed off the coast of Long Beach, got pulled into the port, and then driven up in a car to Stanford, where the PIs were finishing up the work. So those were definitely some well-traveled cultures.
Was it one 6-well or multiple?
Multiple 6-well
plates. And how big is your CO2 incubator that you?
So you can fit, you could probably fit 24 6-well plates in there.
Okay.
It's a pretty big size.
You didn't try to split these at any point, right?
No, we didn't. That's its potential for future work. I think there wouldn't be a technical reason why you couldn't do that.
I was thinking, I listened to an interview you did with Stanford while you were up there, and you were talking about cell culture. Now, if you tried to plate out cells up there, they would never sit down on plastic, right?
Yeah, they might not adhere. So you can look at, if you want the cells to have some kind of connection with each other, what we do in the bioreactors in the ground is seed them with some type of bead, where you can actually nucleate some cell growth. And so that's a possibility. But you may also want to look at cells with no adherence that are just in free float.
Because you would think if you put cells and they're floating in medium, and you have a membrane on top to restrict, so the medium is not just floating, right? The cells would start to divide, 2, 4, and the question is, how big would they get before the ones in the middle couldn't get nutrients anymore? It's kind of an interesting
question, and what does that structure do when it's in free fall?
A lot of cells aren't going to divide if they can't adhere. I can imagine them adhering, finding some place to adhere.
Just randomly bump into the plastic and stick, because that's true. Only a few cell lines can be adapted to spinner, right? So that's an interesting experiment to do. And I don't know, the myocytes probably don't like to be in suspension.
Yeah, and I think looking at the suspension cultures, it's very interesting because in the spinners, they're always subject to a fair amount of shear force. And so on orbit, it's this very gentle process where the entire vessel, the entire space vehicle, and the experimenter are all in free fall, so they're not with that constant fluid shear
So
force.
in terms of long-term motivation for space exploration, is there a motivation for culturing cells in space?
Well, I think it's part of the basic research package. You know, the motivation for space exploration is manyfold. It's our desire to explore. You know, we want to push into the solar system. We want to go to Mars. We want to go beyond Earth. But we have this incredible research laboratory in the space station. And I think there's probably a fair amount of cellular biology, I think also human physiology that we can discover. We haven't evolved to be in microgravity. We've been in 1G our entire developmental and evolutionary history. And so when you all of a sudden take humans and then put them in microgravity, very interesting things happen. You get this massive fluid shift upwards. We're seeing some interesting things that happen to astronaut vision changes on board. We're looking at brain structure and brain changes, increased pressure behind the eyeballs. We also have a high exposure to a CO2 environment on board that we don't have on Earth. We have things like a closed loop environmental system. So we recycle, it's over 90% at this point, we recycle all of our air and our water. And this is a fascinating environment when you really leave the planet and you're supporting human life.
Yeah.
It's a pretty big place for
So
discovery.
it's a great model for going to other planets to see if you can establish this in space initially.
Absolutely.
Close to home. If you get in trouble, you can always go back.
Yeah, well, and you think that cultured cells are a pain to take care of. Humans are very, very difficult to take care of in space. They need all this air and water and CO2 scrubbing.
Yeah.
And
Now you said you had a microscope up there, is that right?
yeah, so we had a video microscopy.
And did the cells divide in the course of the experiment?
I have to leave it to the researchers and all of their data analysis. I think they were probably not dividing too much. We didn't see huge overgrowth, but I think that you certainly could have a dividing cell line up there. Splitting cells, I think you'd need to work out the technical logistics of that a little bit.
If you wanted to do it, you could do it, I'm sure.
You could. I think it'd be fascinating, for example, to have a chemostat up there and look at some, yeast over generations.
From my perspective, it would be cool to infect them with a virus.
I was going to say, next trip, she's got to infect them, right?
Or do a plaque assay.
Right.
You could do the first plaque assay in space. Wow, that would really be impressive. That would be a challenge, but if you don't try, you don't overcome it.
Absolutely.
So what else did you take up? You took up your own little laboratory, right?
So I took up a lot of just pipettes, tips, tubes. I was interested in the things that we use in a normal lab. We do our lab work. You've got fluid in a 96 well plate or in a conical, and you move it around with a pipette. And there's kind of been a lot of canonical thinking that that's not going to work in space. Everybody says, well, it's going to fly out of the tube. And I thought, I just based on the work that I've done, I think that you can probably use the tubes or the tips to contain most fluids pretty well. And I brought up everything trying to break it and try to figure out where does this not work. And I was actually incredibly surprised. You can put fluid in a 6 well plate and turn it upside down. It doesn't matter in microgravity. It has no change in orientation. And it's held in these vessels by the surface tension. Surface tension becomes incredibly dominant for fluids when you don't have the force of gravity.
Is there a limit of volume that the surface tension will act on?
I went up to about a 50 mil conical and could turn those things upside down, no problem.
So you could actually be doing 10 ml pipetting and it would work.
I think it would be fine.
So let me get this. You have let's say you have a 50 ml conical and a 10 ml pipette with a pipetter, and you put it in and it will stay in the tube because of surface tension.
It will, and then when you dispense the pipette, I never noticed the sort of gravitational chauvinism I've had, but I've always dispensed at the top of a tube and then expect that gravity will take the fluid down. That's not a good idea in space because it actually nucleates a bubble, comes out, touches the other wall of the tube, and then you've got a big air bubble on the bottom.
I remember this video of Chris Hadfield wringing out a washcloth that was full of water, and the water crawling up his arm.
It's kind of a horror movie, isn't it?
Yeah.
It's a little bit unsettling,
So
actually.
you put the pipette all the way in the bottom of the tube.
So you put it, just off the bottom, and when you dispense it, you actually dispense a little bit slower than you would on Earth, and you can fill up the tube from the bottom without getting an air bubble in there.
So, this sounds trivial, but all this is new, and you're figuring out how to do all this up there,
I just put a video camera
so
on
you've
on the lab
recorded
bench.
all this as well. Oh, that's just great.
So the big headline was you sequenced DNA in space.
Yes,
right?
that was also a tech demo experiment, but it's something that we're really interested in long term, as we move further and farther from the planet, we're not going to have the ability to send samples back. So the way we do microbiology right now on orbit is we'll take samples, we'll streak a plate, or we'll take a plate and push it against the surface. We do this only right when a Soyuz vehicle is ready to leave. We then package those up, we send them down on a Soyuz, it lands in Kazakhstan, it gets picked up, delivered to the lab at JSC, and then assayed for any clinical microbiology. And so this, you don't get your answer very, very close to when you're taking the sample. And so one of our questions was, could we be developing sequencing technology so that potentially you could, down the road, start to use this for either microbial analysis, or maybe you're looking at some marker of bone loss, and you want to assay that real time. You want that time course of astronauts on board.
So you said you streak plates up there. So obviously the plates are delivered to you, right?
Yep.
And it's a regular sized agar plate.
Yep.
So you could make agar art in space.
Yes, you could make agar art in space.
That would be another first.
I'm just thinking, if you had a bunch of plates on a craft that you're going to send to the space station, they better be oriented right there. How many G's do you experience taking on?
You experience 3 or 4 G's, yeah. So it's very particular, the packing orientation, and then also these plates land. They go back through the atmosphere like we do as well.
So you got to orient them right.
Yes, you absolutely do. And it's a little bit harder when everything's floating. There's a little bit of learning about how to have a good sterile technique on orbit. So on the one hand, it's a little bit easier. You can just put it out there and let it float. And you're less likely to have something raining down on it. On the other hand, you can't manage more than the plate and the lid and maybe one other thing. So actually trying to figure out how to keep everything sterile when they're all floating.
So you can open the plate with one hand, right? And but you don't flame your loop, right?
No.
Well, how do you sterilize? You use a prepackaged loop?
Prepackaged, yeah.
Plastic loop.
Yeah.
Okay.
So my technique was to take the lid off, float the plate, float the lid, grab the plate, streak it real quick, get the lid back on, and get the whole thing taped up.
That's great. Love it.
So what did you use for a DNA sequencing apparatus?
So we actually used a nanopore-based sequencer. So the big machines that need CCD cameras and lasers and those kinds of things will probably not be adaptable to space. They're really large. You'd have difficulty getting them adjusted to launch loads, and so it really hasn't been something that we've been able to take to a remote environment. So it's really the development of the small portable sequencers that lets us adapt something to launch. And we started out first with prep libraries. We launched multiple aliquots of prep libraries and then compared on-orbit sequencing to the ground-based sequencing. And recently we've evolved this experiment to actually streak a plate, grow up a colony, isolate that, and then prep the libraries on orbit, and then sequence them as well. Peggy Whitson just did that experiment.
So did you have to adapt the machine at all to work in space?
So we did very little adaptation. We actually have a lot of work these days at NASA to just take commercial equipment and be able to fly that on board. So we flew a tablet up, and we have an inverter. You used to not really be able to plug in anything with the prong equipment on board space. All of our electricity is from the solar arrays and is managed and needs a DC pin and socket kind of configurations. But we've got an inverter now, so we plug the sequencer in, we plug the tablet in, and the only adaptation I really had to make was that when I was loading the sequencing reaction, you're putting a fair amount of force of your pipette to load it, and every reaction has an equal and opposite reaction. When I first did that, I flew away from the sequencer in the opposite direction. So I went and brought a couple of little foot rails that I could stabilize myself with, and hooked into the foot rails and kind of offset that pipetting force with my feet, which is how you do everything on board.
So that'll go into the manual, right?
Yes. You must hook your feet in before you add anything to the sequencer.
So how big is the sequencer that we're talking
so
about?
It's half the size of your cell phone.
Yeah, it's one of those tiny minion type ones. You put a sample in, and then it does the reaction.
Yep.
And then the data get loaded somewhere. Where does that go?
Yeah, the data comes down in real time, and so it's actually nice. You can see once it does the pore check, you can see the data coming through, and then the ground actually there's other satellites in orbit. We've got a TDRS satellite network. So it goes to the TDRS network, it gets beamed down to White Sands facility, and then it goes to Huntsville, which is our data operations center, and then gets distributed. So they can pull the sequence files down. We're actually looking at ways now that we could put databases on board and actually do some of the computational work on board so that you're completely bypassing that ground step.
So did it all work? Did you get the right sequence?
It did. You know, I was, like I said, I was extremely nervous about loading this first reaction because, again, it's if you screw this up, it's not like we're launching another rocket tomorrow with some more libraries on it. So I had every tool at my disposal. I had magnifying glasses and a light and all of that, and it came through, and there was a lot of cheering on the ground. It worked right away. We got great data even out of our first run, and so we had a bunch of replicates just in case things didn't work out right, and we actually used those for some different experiments looking at how long the flow cells would last in microgravity. Could we reuse the flow cells? What was our fidelity if we're doing 3 or 4 different library preps?
So you brought up a preparation of DNA to do this on,
Yes,
right?
yeah.
But and you mentioned taking a colony and extracting DNA there and trying it. Have you tried that yet?
So Peggy Whitson she's still on board the space station,
Okay.
so she's just done that very recently.
Wow, that's amazing. So in the future, you'll be able to actually look at the internal microbiome of the space station, because you talked about that on your video also. Tell us a little bit about that.
Yeah, I think it's a fascinating environment, and we haven't we sample certain places in the spacecraft for our environmental monitoring, but again, that's all culture based. And we've just started to look at a few time points during the course of an expedition. But I'm very interested to learn what the steady state microbiome of the space station is.
Right.
So is this something that's it's a closed loop environment? Is it very robust and solid, or is it there are a lot of alterations when you inject new humans into this system?
Sure.
And what happens really in our water loops and in our air loops? How variable is that? What happens to the microbiomes of the crew members? So we're in quarantine when we launch in Kazakhstan, but then we get up there and we're completely separated from the rest of humanity. Does our microbiome change? Do we start sharing microbiomes?
Yeah,
How does that influence by the spacecraft? How do we influence new hardware that gets launched? Could you have something? It's not really sterilely processed, but it's going to be pretty low count.
Sure.
How do we populate that, and what happens to that?
So the idea would be to swab different areas, and then you'd have the capacity to sequence, do some amplification.
Yep.
Do you have a PCR machine up there?
So we do. We have a couple different PCR machines. We've got a couple real-time PCR machines.
Better than my lab,
And then, and we've got a real small PCR machine that was actually sent up as part of a student competition, Genes in Space. So there's a mini PCR on board. A couple different ways that we've been looking actually to use as isothermal holds as well.
Neat. And it's being done now, taking a colony and isolating the DNA. But the idea would be to swab different areas, amplify, and then sequence. You could be generating data and publishing it. You can
Absolutely.
Maybe submit it from space even.
So the air is constant. It must be HEPA filtered.
It
right?
is. It's HEPA filtered, and one of our biggest concerns when you're keeping humans alive in this closed environment is CO2. So that's a huge problem. We have ways to remove CO2, but some of it is not very efficient, and it's this really large hardware that will actually absorb the CO2 and then bake it off in heat cycles. And that's one of the things that NASA is very interested in, is how can we better be capturing the CO2 from our environment. We're starting to learn that high levels of CO2 are pretty damaging long-term for humans. It actually impairs memory, concentration. It can give people headaches. How this interplays with the fluid shifts, we're not sure. We're starting to learn about that. But we do know that we really need to be getting the levels of CO2 in our spacecraft down to a lower level.
And this could also influence the microbiome as well,
Absolutely.
right? Because the atmosphere is completely different from that on Earth.
It is. It's also very controlled, which is nice. And you have some very nice willing human subjects who are extremely well profiled and generally biddable. And the other thing is that we have a food, a diet that is completely controlled. It's not, it's generally irradiated, or the microbial count is kept low, but we're not sending up completely sterile food. But we do have very specific space packaged food, which is going to have a strong influence on the microbiome.
So I was just wondering if you do the typical high school experiment of just taking a plate and take the cover off and leave it out for a while, what do you get? Do you grow stuff?
We actually don't get a lot of counts at all from our air samples. And part of this may be the fact that it's very filtered. It's also very dry up there. We do sometimes see, when we do the surface tests, you will see higher counts, but we don't, I would say in general, places that are dry on ISS are pretty low microbial counts. There's some very interesting places to sample where our water loop or our condensate loop is that are constantly wet, and that we've got some interesting selection pressures there. We're in this iodine loop on the US side or a silver loop on the Russian side.
Are these HEPA filters ever changed out and brought back to Earth?
They are changed out, and this is like the microbiologist dream, and we're always asking them, hey, can we get stuff from the filters? And I think there's been a few small studies that have looked at that. But even one of the things that you notice in space is actually the dust buildup on these filters. The airflow is so strong, and any particulate that might come off your clothing, human skin cells, everything like that, because it doesn't settle to the floor, it's in the air and it gets sucked into these filters.
Right.
So when we go around and we do our housekeeping, even on a weekly basis, you'll see a 1/4 inch of dust building up on these filters. And of course, I look at that and I go, well,
it's your skin, right?
we should be sampling this. You know, we should be taking some samples here.
So on Earth, when the sun goes through a window, you can see particles suspended. Is it a similar thing?
It is, yeah. There's a lot of particulate,
Right.
but it's also moved around. The spacecraft will push that airflow very highly. We don't have natural convection on board, and so you actually have to push the airflow pretty strongly to avoid building up pockets of CO2.
Yeah.
Or when we introduce
Interesting.
oxygen into the spacecraft, we want to get that distributed very
So
quickly.
there are no blowers that are circulating air?
There are, yeah. There are blowers
Okay.
that are moving the air through the cleaning system, and then as we're introducing oxygen or we're scrubbing CO2,
Okay.
they're moving it in the air
Okay.
handling system.
How is the oxygen generated, by the way?
So we split water.
Okay.
It's a great system on board. They even have a Sabatier reactor, so they're working on reclaiming
Okay.
hydrogen as well. And
You must have read The Martian, right?
Yes.
Is it pretty accurate?
that's actually one of my most similar. I'm not sure that we're going to have the logistics support anytime in the near future to set up a huge base like that, but these are the kinds of things we would be thinking about doing.
Yeah. Now, the other variable is all the radiation that's coming through, which can affect the microbiome, right?
Yes.
And be really different from on Earth.
Yes.
So it'd be interesting to see how that, I don't know how you can control for it, but that's part of it.
Yeah, and I think the low Earth radiation environment, we do have a radiation environment there. We're still a bit shielded from the Earth's magnetic fields based on where we are in orbit. When we start doing things like going into lunar orbit or going to Mars, we're going to experience much more radiation. And so understanding what radiation does, for example, if we ever want to use synthetic biology for some of these carbon capture or for example, if you want to process waste, we need to make sure that when we're subjecting our bugs to high doses of radiation, that we're not changing their capabilities.
Now, what about the exterior of the space station? We assume it's sterile, but is it?
It should be mostly sterile. Having been out there, it's very harsh. It's exposed to the vacuum of space. It's minus 200 Celsius in the cold and plus 200 in the heat. There have been some astrobiology experiments looking at things like spores, but really long-term survival on the outside of the space station is I would say, in the very, very slim to none, unless you're an extremely hardy microorganism.
But we
Take
know
any swabs?
Yeah,
no, that would be a great experiment, though.
You don't know till you do the
bacteria
experiment.
are pretty adaptable, as we know. They live in really extreme environments.
They are.
And so, I presume you've never brought up any archaea, just bacteria, right?
There have been experiments that have small container type systems that are looking at, and they're just looking at gene expression and adaptability. But they're always completely contained and separate from the rest of space station.
So of course, the other thing that's interesting is when you come back down, you look at your microbiome and how it changes and readapts to wherever, because you go back to a normal life, eating regular food that you really like, right?
Absolutely. Yeah.
It's going to change. Nobody's looked at that yet, though, right?
They've just started to do a few time points, but we haven't done a really in-depth, detailed time course of that.
So I'm interested in the virome as well.
Yes.
And is anyone thinking about that?
Yes, there are few people are, because I've cornered them at conferences and said we need to do this study. So there's been early studies that have been indicative of latent virus reactivation, and there's certainly medical case reports about maybe having more cases of shingles than you would expect.
I got a friend, Dave Bloom, who has he's had a NASA grant to look at HSV in space and in astronauts.
Yeah, and I think looking at some accessible tissues and profiling the virome of astronauts. I mean, we're just starting to learn when we do deep sequencing about what the normal virome is of humans. And the question is, if you're potentially slightly immunosuppressed, we do think that astronauts have some degree of immunosuppression, and you're in this microgravity and very stressful environment, what happens to your standard virome?
So presumably that's what's reactivating latent herpes, the stress of being up there and all of it, right?
I think it's early days, but I think it would be a really fun and interesting area to explore.
And again, you could develop the capability to do it all up there and be independent of sending things back down to Earth, right?
Yes.
Is that one of your goals?
Right now, I just want to start taking the samples, and so we're looking at, we have some astronaut repository samples where we take samples pre-flight and then follow people during their mission. But a lot of those have been cell-free samples or sort of limited sample types, and so I'm looking at expanding the kinds of samples that we take, looking at more saliva samples and cellular material.
So when did your voyage end?
I landed in October in Kazakhstan, 2016.
And so what do you do now?
So right now I'm the Deputy Director of Human Health and Performance. So this is all of our, it's our environmental laboratories, our microbiology lab, our flight surgeons. It's about 900 people. Anybody who's involved in coming up with requirements for new spacecraft or figuring out how we're going to support humans and further exploration, as well as supporting current station programs.
You get another trip?
I hope so. You have to ask my boss. The chief of the astronaut office assigns people to a mission.
So it could be that you would not get another trip?
Potentially.
Wow.
Yeah. You always assume you've got your one time in space and that's it.
Wow.
I never take a day up there for granted.
That's interesting. Well, in the meantime, you can plan experiments that will be done by others in space.
Yes.
And you said some are ongoing right now. Are there any of them you can tell us about?
Yeah, so there's a lot of really interesting experiments that are looking at expanding our cell culture capability. There's an osteoblast experiment that's going on right now. The sampling experiment I told you about. There's a lot of plans for what we're going to do to expand on the twin study. So that was the Mark and Scott Kelly study that was a number of looking really at epigenome, transcriptome responses to spaceflight, how we're going to develop a full picture. I'm interested in what we can do to better understand the physiology of humans on board. This fluid shifting thing is very strange. I can tell you it feels very weird to all of a sudden not have that gravitational pull of fluid into your legs, and that's one thing that doesn't adapt over time. So you sort of always have this stuffy head, and fluid is moving into compartments that we haven't evolved to handle, and I think that's having some secondary effects in terms of physiology over the long term. That's really interesting.
And this is something you feel immediately as soon as you get into the...
You feel it as soon as you get into orbit, and it's one of the... there's some neurovestibular issues, for example, or disorientation that will go away after a few days. Your brain is incredibly adaptable. You learn to locomote, and instead of walking, you fly, or you hold on to a handrail. You learn how to stabilize, but the fluid shift effects will stay with people for the whole mission.
So when you come back, is it a significant adjustment?
It is, yeah. I came back, and I think one of my quotes in the gym is, "What am I doing stuck in this 1G hell?" It's amazing. You've never noticed gravity in your entire life until you spend a few weeks without it, and then it was things like picking up my iPhone and just having it sort of fall onto the table because I wasn't used to holding the weight of the iPhone, or having food slide off my fork because you put your fork into food on board and you can put it in any orientation that you want. So
I've always thought that a hard landing in a Soyuz after being weightless for 3 months would be something.
It's something. The parachute deploy sequence is something as well. I think Scott Kelly said, "In a barrel lit on fire going over Niagara Falls." That's a pretty accurate description. So you get through all that, but there's definitely a period of readaptation into microgravity, and so we actually we take a lot of data, and we're taking a lot of time points as humans are transitioning back to dealing with gravity. And I would say after about 2 weeks or a month, you feel pretty normal again. But it is a fascinating feeling to feel incredibly heavy. So we landed, and part of the experiment for our stabilization is to go do some obstacle course tests, and you can kind of barely stand at that point, and then at one point they tell you to lay face down, and the researcher comes and says, how heavy do you feel compared to pre-flight? And I said, well, I'm at least 300 or 400 pounds, and I'm stuck face down on this planet with magnets. I am being centrifuged onto this planet. I cannot push myself up and get off. They said, don't worry, you'll be fine in a week or so.
So this fluid shift that you mentioned, that also takes a few weeks to
It does,
to go away?
yeah. It takes a couple days, and you can definitely tell that the body's going through this rapid physiological process to readapt to gravity.
One of the things you mentioned in your video is that every astronaut gets trained to do experiments. And why?
Yes.
Why is you want everyone up there to be able to do something?
Yeah, because we've got a long, sort of pipeline training for astronauts. The astronaut launches then need to line up with our cargo launches, so we're not exactly sure what experiment is getting launched when, and we want to make sure that we can launch. We did 275 experiments when I was up there, and I was hands-on with about 100 of them. So we want to make sure that we can launch things and have somebody able at any point to carry out that experiment. Now, you're not completely on your own. So you have the comm loops and you have video loops. And so oftentimes the PI has this connection on board, and they're walking you through things, and you have procedures written out for you. And you can really train people to do any kind of bench work that you need them to.
Yeah. So I forgot my question now. What was it? You got one?
What was, well, I got 1,000.
I know. You could be here for hours, right?
What was the coolest thing?
I was a little bit cynical before I launched about the planet. I said, well, yeah, I've seen that on TV and movies, and it's just not going to be that impressive. I've worked in mission control and seen that a lot. And we launched, and I saw the planet out of the window of my little Soyuz. Now, we were in the Soyuz for 2 days before we docked to the space station. It was this tiny, tiny little spacecraft. And the first time I saw the planet, I couldn't even talk for a good 5 minutes afterwards. It's amazing. It is. It's much bigger and brighter than you could imagine. There's something about when we capture it on film, it doesn't have this glowing effect, but you actually really see the Earth as a planet suspended in the universe. I mean, you can really tell that you're orbiting the Earth, and the Earth is this planet, and it is much brighter and far more majestic than I ever could have pictured.
Is it the most beautiful thing you've ever seen?
It is the most beautiful thing you've ever seen, and you never get used to that. I mean, even after 4 months, I would still, finish work for the day, and we've got a cupola. We've got some windows that overlook the Earth, and you can actually get your whole body down into the cupola if you curl up, and so you can fly over the planet. And so I would fly over continents for 90 minutes, 2 hours, and watch day cycles and night cycles. You fly across the entire continent of Africa and just watch thunderstorms, and then you go up and you head over to Europe and Turkey, and then before you know it, you're floating over the Sea of Japan. And so just to watch the natural phenomena on our planet through all these day-night cycles is amazing.
And you can see lights on in cities as well.
You can
right?
see lights in cities. You can see massive fishing boats actually in Southeast Asia. You can see lights from that. You can, with the long lens of a camera, you can get down and see kind of individual houses on the road if you're looking through an 800 millimeter lens.
I would think that a spacewalk would be awesome.
It is, although there's a good quote in our office, anybody that says spacewalking is fun hasn't done one, because it's we call it type 2 fun. It's very fun after the fact. When you're in the middle of it, it's one of the more ridiculous, outrageous things that human beings do. And you're very focused. It's pretty amazing, but it's also 6.5 hours in your own little spacecraft, which has a limited amount of consumables. It's very risky. You really increase your risk level when you're out there. And you're hanging on to this structure, and you're going 17,000 miles an hour, and you are not contained inside a structure anymore. And so everything is very carefully thought out. You must tether to your tools and equipment. You must tether yourself to the space station all the time. You don't want to all of a sudden become another Sputnik, be a satellite.
So if you unhooked, you would just float away.
Yeah, you would float away if you put in any little reaction force. So we're tethered in multiple ways. We've got a lot of safety protocols, but it's pretty high in the back of your mind.
Type 2 fun, I like that.
Type 2
That's
fun.
good.
Yeah, that's great. So how long is the career of an astronaut? How many years? So you could be on the ground, I guess, for as long as you want, right?
Yeah. So most people tend to be in the office for anywhere between
the office.
10,
I
15,
love the office.
10, 15, 20 years. So we used to have, our launch rate used to be a lot higher with with shuttle, we're launching people a little less frequently, but for longer duration now. So our missions are these real long duration, more than 100-day missions these days.
So speaking of launching, are we going to develop launch vehicles again?
We are, yeah. That's actually one of the things that we're working with from the certification point of view, is a couple of different U.S. companies actually have contracts to provide commercial launch services. So SpaceX, Boeing, and Sierra Nevada are all working on launching humans, very shortly. We're going to start providing that. So we've been doing cargo launches for a long time from the U.S., but we want that capability to launch humans again.
Right. Is there a age limit beyond which you can no longer go into space?
There's not a hard age limit. Probably around your mid to late 50s, you might not be able to keep your medical clearance.
I see. But John Glenn went up at the end, right?
Yes.
How old was he?
So he was a special case.
Yeah,
that was a one-time flight. But astronauts have flown in their mid to late 50s.
So what do you want to do? Do you want to keep going up as long as you can?
Yeah, you never I think lose that allure of space flight, and once you've done it once, there's nothing more amazing than looking at the planet. I think there's a lot of work to be done up there experimentally. So
sounds like it. Yeah.
I want to go up and do all the experiments and do all the science. I never had trouble with my crew members. We get these things that come into our timeline, and they said, "All right, Kate, you got all of the experiments if you want them."
So you'll hang with us for as long as you can.
Absolutely.
Okay.
So I don't hear you going back to lab work on the ground anymore, right?
Well, I would, I'd love to do lab work on the ground, and part of that is combining, some of the work that we're doing for payload development. How are we going to get higher throughput science done on orbit? And I think having that experience of actually having handled things in microgravity, it helps us work with a lot of these different experimental, we call them payloads, these payload developers.
Now I remember my question. It takes 10 minutes. You mentioned you were going to publish some of your findings on that video. Is that still something you might do?
We've actually submitted a few papers.
Cool,
so
nice.
I think we're getting reviews back. And
Maybe we could do them on
Yeah,
TWIB.
we'll do them on TWIB, absolutely.
Well, they're microbiome papers, right?
We've got some microbiome papers, we've got some sequencing papers.
I have to do them on TWIM. My TWIM co-host's there. Now, are we going to Mars?
We are going to Mars. We have a lot of questions to answer before we go to Mars. Mars is hard. But Mars is fascinating. It really drives us to push our comfort level in terms of technology and engineering for keeping humans alive. And it really changes our thought process in terms of Mars is going to be an autonomous, independent expedition of people leaving Earth. Right now, we're very dependent on low Earth orbit. We have all these nice logistics suppliers. We can send our plates down. What we think about in terms of our medical capability, food production, independent sustained living, scrubbing CO2, the reliability of our life support control completely changes when you put Mars into the equation.
Some of these things
go
you
to
can.
Mars?
Yeah.
I would go, if I had good crewmates, I would go to Mars. It's going to be a long journey. It's
Crewmates.
2.5 years.
And it's a one-way trip.
No, it's
could
a
come
round
back?
trip.
You can
It's a
come
round
back.
trip. Yes.
So if you landed and established, you can take off again.
Yes.
Okay.
Yeah.
I always just thought it was one way.
No,
So-
NASA's architecture involves a return to Earth.
So with the right crew, you would do that. You could have a lab there and establish it and so
Yeah,
forth.
I mean, it's something we're not going to be doing for a while. It's mid-2030s, but it certainly is what we're thinking about right now. We are designing spacecraft. We're thinking about what you need to do for the life support system.
Yeah. Now, let's talk just briefly about life out there. Are we the only ones, or is there something else?
So I would say that we're likely to not be the only ones, but the question is, when would we coincide in time and space with other life? And I'd put a huge bet on microbial life, right? That's the most likely. But the universe is vast, and the universe has been around for a long time. And so if you think of what's our life, of life evolving right at this moment that we happen to be here, I think might be slim, and it depends on how abundant it is in terms of if we're going to ever bump into it.
So it seems to me that when people talk about life other than on Earth, the assumption is made that it's nucleic acid based. And that strikes me as not necessarily true.
Yeah.
So if you want to discover life elsewhere, and you don't make that assumption, how do you define life? Do you have any thoughts on this?
I agree that to say that it's going to be nucleic acid based is an Earth-centric point of view. I think what we need to do in terms of technology is have a wide suite of tools that's able to detect things. And so you're going to want to be looking for chemical signatures, and you don't necessarily want to be looking for A, T, G, and C. So you want to be looking at modified bases, at bases that aren't necessarily carbon-based. And I think we have a pretty good set of scientific criteria to determine if things are alive or not. And we have methods, but we want to be developing multiple different methods. So we're looking at the sequencing, particularly because it may be able to detect modified bases. But you'd also look at spectroscopy and signatures.
Yeah, it
of
seems to me a
chemical
mass
products.
spec would be one of your main tools.
Absolutely.
You want to look for molecules that aren't going to happen in just spontaneous ones,
Absolutely.
right?
There's
Okay.
no plans to bring a mass spec up to the space station?
So we actually, we do have some mass spec technology that does our atmospheric composition, and I'm sure there are instruments that are being developed. Whether or not they're the same ones that you would put on a spacecraft that's going and looking for life detection on an Enceladus or something, I'm not sure that we're testing those on ISS, but it's a great idea.
Now, we're probably going to go back to the moon, right?
That is in the current architecture plan to have a gateway architecture for lunar orbit.
And I bet you'd like to do that also,
I would
right?
I would volunteer anything that's leaving the planet. I think that's a.
What about beyond Mars? Is that ever going to happen?
I think Mars is the hard challenge that is within our technology capability right now. We'd have to push on a lot of things, but it is doable. Beyond that gets into we're relying on technology that hasn't been invented yet.
Do we have any online questions? Yep.
Okay, so we have a question from Ruta, who is watching from India, where it's currently about 1 a.m., and he'd like to know, what are the chances of us contaminating other planets in the solar system as we explore them with spacecrafts like Cassini and Juno? And he also asked about the significance of sequencing DNA in space.
So that is a great question. We actually have a planetary protection office at NASA that really thinks about this. So for example, when we send a spacecraft, when we're looking at Mars landing site, we carefully assess spacecraft cleanliness so that we're not going and contaminating. And there's actually sites on Mars that are special protection regions. So for example, if there's something that may be near a water environment, they're actually going to go to extra precautions. We deal with this even things like our spacesuits. So our spacesuits are not sterile. We have some leakage rate, and humans leak bacteria and skin cells and all kinds of things. So we actually would think about any kind of exploration suit, how we protect the planetary atmosphere as well. So it's a strong area of interest.
And the second question was, he was wanting to know the significance of sequencing DNA in space.
Yeah, so part of it was just a technology demonstration to show that this technology works. We weren't sure if nanopore sequencing was going to work on board, if there could be bubbles introduced that would, bubbles in your microgravity system will kill you. They're all over the place. It's hard to get them out of the system. They form micro bubbles, and so we were wondering, could we do this in a bubble-free environment? We had a pretty good idea that it's going to work based on capillary action and sort of these microfluidics principles, but we didn't know for sure, and we were able to demonstrate end-to-end that we got as good, if not better, results on board than we did with the sequencer on the planet.
I do. Another question from online do you have any shareable results from your space experiments, and also what was your favorite experiment?
Yes, so results are forthcoming. I don't want to scoop anybody's publication.
You're not in danger of getting scooped from space, are you?
Right.
Well, these are experiments that I participate in, but this is not my research. I'm not the PI on it. So, but we do have a lot of things that NASA publishes, anything that we fund. And some of the things I think that are coming out are starting to be the molecular capabilities of the space station. And so I can tell you, we can do long-term cell culture in space. We can do sequencing in space. We did a couple of experiments to demonstrate real-time PCR, quantitative PCR works just as well on orbit as it does on board. So I can't say that the capabilities are there in an Earth as you would have in an Earth-based laboratory. It is 10 to 100x more complicated to launch an experiment from the planet, but it is absolutely doable.
A couple of years ago, it was attempted to make protein crystals in space. What was the outcome of that?
So that's still ongoing. They actually, there's Japanese and European experiments, and there's some Russian involvement in the European experiments, and so they're looking at growing crystals for a number of different proteins. But one thing that you have with a space experiment, it's got to be very carefully designed, right? So somebody who's doing crystallization is going to be constantly tweaking, and adding things. And so you have to have, I think, a pretty good idea of what you want in your crystallization medium. But that being said, you have potential to develop crystals there that are going to be a little bit larger and a little bit better than you could on Earth.
I just wanted to correct myself. Ruta is actually a woman. And do we have any questions in the audience?
Could your work be affected by the budget proposed by the new administration?
So we have a strong support for the space station, and we always have. And so there's not huge changes in the space station research endeavor. We actually have it's a really long-term plan in terms of how we make agreements with our international partner countries. And so that plan has been pretty unaffected. We are thinking about what we're doing with exploration plans right now. And so that I know will be an international effort as well. And we're really starting, at this point is the point in the process where we're starting to get serious about developing the technology and the infrastructure to go beyond low Earth orbit. And so that's going to be something that starts to get developed over the next few years. But space station has been agreed to by all countries till 2024. And we have a really robust infrastructure in low Earth orbit to support that.
Thank you. Michael Schmidt from TWIM. In terms of the fluid shift, I find this absolutely fascinating in the sense that when some people fly, they get the fluid shift into their head, and then for days after, they get an upper respiratory sinus-like thing. So in space, do you have an upper respiratory sinus headache for 115 days?
So you don't have a headache the whole time, but we certainly do have increased reports of headaches. And you just have this this sort of head full feeling. So if you lay down on your bed or your couch and you hang your head off the end of it for a few minutes, that's about what it feels like to be in space. And that doesn't ever really go away. You just get used to it, and it becomes just part of the daily environment. So we don't have increased incidence of infections up there because we're in this nice little isolated environment with no other pathogen producers.
That brings up an interesting question. If you were getting ready to launch and all of a sudden you came down with a cold, would you be scrubbed?
So you probably wouldn't be scrubbed. You might be delayed. We are in quarantine.
Okay.
So we have a progressive quarantine before launch, and we've always maintained, when we launch from the US or we launch from Kazakhstan, we have this about 2-week quarantine. Now that's not to say somebody couldn't develop an illness right before launch, but we're definitely taking a lot of precautions to reduce that.
People do get sick in space, they must, right?
So you don't, I didn't get a cold the entire time. I mean, I'm up there with 2 other people or 5 other people. And if they've been doing the quarantine right, the new crew should come up disease-free.
Yeah, so the big deal is the latent infections.
It is, and it's also the physiological effects from things like CO2 or fluid shifting. That's what provides you your physiology challenge. But it was pretty nice. I came back and landed on Earth and went to an airport, and I was like, oh my God, this is, there's all these people and all their viruses down here on the planet.
We have one more question here.
Thank you for being here. Teja from University of Illinois at Chicago. My question is, do you get checked for non-symptomatic HSV viral shedding before you get onto the space station? I mean, there could be a lot of viruses that you'd be shedding, right? I mean, do they check for the rate at which you're shedding or something?
So we'll do some normal clinical screening of all the astronauts. So we'll do a normal workup that you would get. We do check people for MRSA before they go up there. You know, and you know, we'll look for things like H. pylori and that sort of thing. But in terms of just kind of standard virus shedding, they're not too worried about that. It might be an area of interest, and if we find that we're getting a lot of latent virus reactivation, I would imagine we're going to start looking at that more closely.
I have two questions, one serious, the other less so. The serious question is, surely developmental studies have been done on development of plants and animals, and is there evidence that gravity affects them? And my second question is, what do you do for fun?
Yes, great question. So the first one, they've actually done a lot of plant studies looking at how plants are responding to the lack of a gravitational vector. We did a few Arabidopsis seedling experiments that were actually growing seedlings either in a centrifuge with the 1G environment or without the 1G environment, and actually looking at plant growth and at seedling growth and at gene expression. And so, you know, light tends to be this really predominant variable, but there are changes in plant growth and development on board. As you can imagine, it's difficult to grow things in dirt when the dirt would float, and so there's actually a lot of subsequent technology development experiments that have developed that. There's been developmental experiments, for example, in Drosophila. There's been fish experiments. You know, now that we have a rodent facility, I think mammalian development experiments would be very fascinating to do on board. I'd like to see those coming out in the future. In terms of fun, my rule was don't do anything in space that you could do on Earth. So, you know, I might watch a TV show or listen to a podcast while running, but in my free time, I was at the window looking at the planet. Just the opportunity to observe the planet, or I was doing experiments inside with fluids and watching behavior of things in free fall, and that was constantly unexpected. Even 4 months in, something would absolutely surprise me about the behavior of fluids because everything is in free fall, and it's not our set pattern to expect what things are going to do in microgravity. So for fun, I was doing an experiment, or I was looking at the planet.
Good answer.
So
One more, yes.
I was actually kind of wondering about fluids also. I run a lot of gels in the lab. I don't know if there's a way you can like load a gel and run it. And also if you have a solution of, say, bacteria or viruses, can you pellet them, and will the pellets stay stuck to the side of the tube?
Yeah, great questions, both of them. So I think we probably wouldn't run a gel on board. We would probably do a microfluidics-based chip assay. I think you could run a gel if it was in a closed container where your buffer was completely enclosed, because you could still apply a current to it. But we'd probably use more of a lab on a chip type method. And for your second question, we can centrifuge on board. There's actually one of my favorite experiments was they didn't want to send a huge centrifuge to space, so they 3D printed a rotor and stuck it on a drill. And that actually works great to get the solutions to the bottom of the tube. So you can pellet things on board, and if there's enough force, you know, if you do a standard pellet where your pellet's stuck to the side of the tube, as long as you don't dislodge it, it would stay stuck there. But if you start to put in a little bit of force, for example, if you had a bead in solution, if you put a little bit of force into that, it's gonna start to float, and it's gonna be in free fall as well.
All right, I'm gonna keep to my promise and let you go after an hour. This has been TWIV 444. You can find it at asm.org/TWIV, Apple Podcasts. And if you like what we do, consider supporting us financially. You can go to microbe.tv/contribute. We have Patreon, PayPal. You can even get t-shirts if you'd like. Everyone, if you have questions or comments, twiv@microbe.tv. My guests today have been my co-host, Rich Condit. Rich Condit is a former professor at the University of Florida in Gainesville, currently residing in Austin, Texas. Thanks for coming over.
This has just been a blast.
You wouldn't have missed it, right?
No, I wouldn't have missed this for anything. And if you ever need to test how an old guy will hold up in space, I volunteer.
We'll get you on the recruitment campaign.
Absolutely.
And Rich came out here because of the generosity of our supporters, so thank all of you for that. Of course, our special guest has been Astro Kate Rubins. Thank you so much.
Absolutely.
And I want to give you a present from the TWIV team, a TWIV t-shirt. There you go. So thank you so much.
That is
really
great.
appreciate
Thank
it.
you.
Good.
I expect
And
to see that on your next ride, right?
now,
Absolutely.
would anyone like a TWIV t-shirt? You would like a TWIV t-shirt. We're going to let Kate throw one to
All right.
you.
I'm gonna try to ping our question askers here.
Yeah, go ahead, go for it. If you want more, come to another live TWIV somewhere. We'll do this again next year. We're going to have a TWIM tomorrow, but we don't have any t-shirts, no t-shirts, but we'll work on that. I'm Vincent Racaniello. You can find me at virology.ws. The introductory music you hear on TWIV is composed and performed by Ronald Jenkins. You can find his work at ronaldjenkins.com. I want to thank the American Society for Microbiology for letting us do TWIV here. It's really a great honor to be able to do this. I want to thank our producer, Chris Kandayan, our cameraman, Ray Ortega. These guys I work with all the time, they're just awesome. And I thank the audience for coming and everybody else online as well. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWIV is viral.
They tell you to put your chin right there.
I became an ASM member in 1981.
95?
Since 2008.
I joined ASM in 1974.
I've been an ASM member for 41 years.
I first became a member in 1989. Seems like all my career.
I'm willing to bet it's a decade.
My first ASM was 1993. I still have a picture of me holding the program open, sitting on the hotel room bed, really excited because for the first time ever I saw my name in print associated with microbiology.
When I was a graduate student at Purdue in 1980, it was the thing. I mean, if you were going to be a microbiologist, you joined the ASM. It was just really that simple.
ASM is very special to me because I became a member in 2002, and when I first attended the meeting at Salt Lake City, Utah, I met my PhD thesis mentor, Dr. Arturo Casadevall. And so ASM kind of introduced me to the PhD world.
Definitely the most important thing is that ASM has provided me with a graduate fellowship. So they've helped support me during my graduate training. In addition, I've gotten a lot of networking opportunities and I've met a lot of really great people through ASM.
And we can share our work and connect, and they can teach me things that I don't know and I can teach them things that they don't know. And just that partnership and working together, that's what scientists do, to share your work. And it's the most exciting thing to learn about new projects.
ASM has actually done a whole lot of work and given me the kind of exposure I wouldn't have had if I'm not a member of ASM.
Many brains are better than one.
Great thing. It's a really member-driven organization. I love that. I've always loved that.
Could make the bigger the better, the more the merrier.
You want to do microbiology? Become a member.